Please watch the whole video. If you find the video educational, please provide the feedback in the comment box, click the like icon, and subscribe the channel. Genes and DNA are fundamental components of life, playing crucial roles in heredity, cell function, and biological diversity. Here's an overview of their structure and function. DNA, deoxyribonucleic acid. Structure. Double helix DNA consists of two long strands forming a double helix. Each strand is composed of a sugar phosphate backbone and nucleotide bases. Nucleotides, the basic units of DNA, consisting of a phosphate group, a sugar molecule, deoxyribose, and a nitrogenous base. The four nitrogenous bases are adenine, A, thymine, T, cytosine, C, and guanine, G. Base pairing: adenine pairs with thymine, AT, and cytosine pairs with guanine, CG, through hydrogen bonds, forming the rungs of the helix. Function. Genetic information storage. DNA holds the genetic blueprint for the development, functioning, growth, and reproduction of all known living organisms and many viruses. Replication. DNA can replicate itself, ensuring genetic information is passed on during cell division. Transcription. DNA is transcribed into messenger RNA, mRNA, which carries the genetic instructions to the ribosomes for protein synthesis. Mutation and variation. DNA mutations can lead to genetic variation, which is essential for evolution and adaptation. Genes. Structure. Segments of DNA. Genes are specific sequences of nucleotide bases on the DNA strand. Coding and non-coding regions. Genes consist of exons, coding regions, that are expressed, and introns, non-coding regions, that are not expressed. Introns are spliced out during mRNA processing. Function. Protein coding. Most genes contain the instructions for building proteins, which perform a majority of cellular functions. Regulatory elements. Some genes encode regulatory RNAs and elements that control the expression of other genes. Heredity. Genes are inherited from parents and determine inherited traits by coding for specific proteins. Phenotypic expression. The proteins coded by genes lead to the development of an organism's physical and biochemical traits. DNA replication. Initiation. Replication begins at specific locations called origins of replication. Elongation. Enzymes like DNA polymerase add nucleotides to the growing DNA strand, complementary to the template strand. Termination. Replication ends when the entire DNA molecule has been copied. Transcription and translation. Transcription. Initiation. RNA polymerase binds to a promoter region on the DNA. Elongation. RNA polymerase synthesizes a single-stranded mRNA molecule complementary to the DNA template. Termination. Transcription ends when RNA polymerase reaches a termination sequence. Translation. Initiation. mRNA binds to the ribosome, and the start codon, AUG, is recognized. Elongation. Trimolecules bring amino acids to the ribosome, where they are added to the growing polypeptide chain. Termination. Translation ends when a stop codon is reached, and the newly synthesized protein is released. Genetic mutations. Point mutations. Changes in a single nucleotide base pair. Insertions and deletions. Addition or loss of nucleotide bases. Chromosomal mutations. Large-scale changes affecting the structure or number of entire chromosomes. Genetic inheritance. Mendelian inheritance. Traits are passed according to Mendel's laws, dominant and recessive alleles. Non-Mendelian inheritance includes incomplete dominance, codominance, and polygenic traits. In summary, DNA and genes are intricately structured molecules that store and transmit genetic information, enabling the diversity of life and the functionality of living organisms. Thanks for watching the video.